In the human side, when we have these nutritional recommendations, they don't say that the processing affects the quality of the food in any way. Right. Like if you read the official guidelines, then you get the idea that like a super fresh food or the same food in an ultra-processed form is actually the same. Right. And that can't be true. Yeah.
Okay, well, there are two big areas that you have to focus on when looking at comparing the nutritional quality of a kibble diet to a raw food diet, and one is definitely the processing, and that the processing doesn't matter. I agree, this is something that is really interesting to study, but the other issue is the actual macronutrient profile of these foods. So like, let's say that you have raw meat, but then you have ultra processed meat that has basically just been well, you've taken the original meat, but then you've cooked it, you've probably pressurized it to
you lose a lot
pathogens.
of vitamins.
You might have added some chemical additives because, well, the meat quality itself might not have been very good, so you might want to supplement with some fatty acids or something like that. The point being just that you can have a absolutely identical diet and then like start deciphering these ultra-processed versus non-processed. Where exactly does the issues lay? But then the other issue of macronutrients is also something we find fascinating, where if you are feeding a non-processed diet, even in a human, it's going to look profoundly different to the ultra-processed macronutrient profile, where if you look at a macronutrient profile of something that's traditionally termed ultra-processed, it's going to have carbohydrate in it, and it's going to have oils in it, which are traditionally not found in non-processed foods with a high probability. So we're really interested in finding what it has to do with, what processing has to do with nutritional quality, nutrient availability, and overall impact on health. And I think that we're just at the beginning of this journey of understanding how to address these issues.
And how to quantify it.
And how to quantify them, yes. But, well, as a scientist, we have to sort of, addressing is quantifying.
Yes.
But, yeah, I mean, yeah, you'd have to look at both of these like separately in order to really determine what it is with processing that really could have problems, but or could cause problems, or might not have that big of a problem.
Yeah, exactly.
But if you would start, it's very interesting to think about whether processing affects the bioavailability of whatever has been put into the original food.
Ah, okay.
So I think that...
how do you measure that? It's probably a long story.
Well, not necessarily. So if you talk about bioavailability,
what does it actually mean? Is it just the absorption, or is there something else to it?
Well, that's a very good question, because absorption is what you would assume bioavailability means. If you get something into your body that, like, I think we could talk about vitamins. Vitamins are a great example, because if you talk about it being available to the body, yes, it can be absorbed.
Yeah,
but
through the intestine, and then it's in your body.
Yeah, but B vitamins are notorious. There are many of them, and they all have many forms. And some of them just might not be able to be used by the body either at all or nearly as well as other forms. That's what we talk about bioactive nutrients and bioactive ingredients, that they're suited for, that basically the nutrient is able to be used by the body as such. And so it's bioavailable, or it can be made bioavailable with very little processing in the body itself.
Yeah, so they don't just get stuck in some receptors or somewhere, but they actually do what they're supposed to do.
Right. So, I mean, the highest likelihood for something that enters the blood system but is not actually a bioactive molecule is for it just to be scavenged up and picked up and treated as garbage. So It would be recycled and it would be taken to the kidneys.
So you wouldn't call that very bioavailable even if it absorbs well?
Right. You wouldn't call that, no, you wouldn't call it bioavailable, and you wouldn't call it bioactive. But so that's definitely, that's a really interesting point. Like, how does processing affect bioavailability?
And different like steps of bioavailability maybe.
Right. And we're able to look at that because we know the concentrations of micronutrients in both of the diets that we've been looking at. So even if we're looking at diets with profoundly different macronutrient profiles, we're also able to say that, okay, well, if we know how much vitamin B6 is in the ultra-processed diet and how much is in the non-processed diet, then we can also assume that we should be getting, if the amount of vitamin B6 is the same, we should be able to see that vitamin B6 activity or vitamin B6 concentrations are similar.
Do you measure them from the serum, from the blood, or even from inside the cells? Or?
Right. Yeah. Just to finish up, however, there's like some problems with looking at like micronutrient concentrations when looking at different macronutrient profiles, because different macronutrient metabolism requires different amounts of vitamins. So that complicates the story, and so it allows us to give more nuanced answers, but then it also allows us to or it doesn't allow us to just give an X, it leads to Y correlation. Excuse me, what was your
I was just thinking that how do you see processing affect the bioavailability of the nutrients in general?
Yeah, okay, so you were looking at, you asked about how are we looking at this, and so what I'm looking at is the metabolism, and so we're looking at blood markers and urine markers. And so your blood is basically a big transportation media. It transports fluids and whatever nutrients that any organ or tissue requires or cell by using a pump and of liquid to just get it to where it's going. And so the blood is a great place to look at for seeing what nutrients are currently being transported, how much, and then we can also hypothesize why. So we're looking at biomarkers for metabolism, which can be found in the blood and in urine. There's interesting reasons for looking at specifically blood and urine. If you look at only blood, then you can get a fairly good snapshot of what's being transported around. But if you include urine, you're also able to look at, okay, this is what's going around in the body, but this is also what the body's actively getting rid of. And so you can, for example, it's been shown that if you see a marked elevation of some biomarker, some metabolite that is high in the blood, but then you also look in urine samples and you see exactly the same metabolite and its concentrations are much higher in the urine as well, then that's a big sign that the body is actively trying to get rid of this nutrient and that there's too much of it. The body is trying to maintain a type of regulation of health all the time, and like it's trying to maintain homeostasis.
Can you at this point also look inside the tissues or like inside the cells whether something is going inside there? Yeah.
So that's I wish it really depends on what you're talking about. For example, some simple signaling molecules are able to, you can draw the correlate that we see this amount in blood, which, okay, so to be clear, when we're talking about metabolites in the blood, then they're not inside cells. Yeah. They're in the serum. Yeah. So we're looking at serum, yes, but it essentially they're traveling freely. And depending on the size of a metabolite, it can enter freely through a cell membrane. So the concentration in the cell should be the same as outside. But then for other metabolites, you have to have a receptor and a protein, like a protein receptor on the membrane of cells that actively transports them into the cell, and then it maintains a concentration in the cell that is different from the serum. So it depends on what you're looking at, and that has to be talked about for sure if you want to then talk about the concentrations of a certain metabolite in the blood or in serum, for sure.
Okay. So the big question, if you would compare, for example, kibble, which is like the other far end, and then like a really high quality raw food, what do you see with your metabolomics?
Yeah, well, that's a great question, and that's what we're trying to find out. We're trying to establish what we see means. So what we see is that there is a big difference in the metabolite profile. Now, we're looking at a targeted metabolite profile. That means that we have tens of thousands of metabolites in our body that are currently activated and active in all types of, well, of course, catabolism and anabolism. It's breaking down things and it's building things, but it's also signaling and regulating these processes. So that's what we think about when we're talking about metabolism, and it includes many, many molecules. Some seem to be more important for signaling processes than others, and then some resemble, there's different categories of metabolites as well. So for the purpose of simplicity, we are only looking at a subset of metabolites. And what this allows us to do is know which metabolites we're looking for. And so when we run a blood sample or a urine sample through a machine that detects these metabolites, by knowing what we're looking for beforehand, we're able to get very precise amounts of these metabolites. We're able to get very precise quantitative results for just what the concentration of a certain metabolite is in the blood or in urine. And what we can do is say that, okay, well, out of the subset of metabolites that we are looking at, we can see that there are very big differences between a dog that has been eating this ultra-processed diet versus the non-processed diet. And we did that by taking blood samples before the diet started, and then we had one group of dogs eating a ultra-processed diet and one group of dogs eating a non-processed diet for an average of 4 months. Some dogs were eating a little bit longer. And then we looked at how this subset of metabolite concentration changed after they had been eating exclusively one or the other diets.
Okay, so what do you see there, and how do you hypothesize, like why does it go that way?
Right. So there's two ways that you can hypothesize, and or let's talk about what we see first. Okay. So what we see is that out of the subset of metabolites that we've been interested in, there are some that increase in the food in the dogs that have been eating ultra-processed foods, and there are some that increase there are some that have been increased in the dogs that are eating non-processed foods. And so you can see that, okay, there's a difference, and it's very significant. And that's a good starting point for then seeing what could this mean, and what could this be because of, and what could it be an indication of. And so that's where you come into this question of how do you interpret it, and what do you do with this data. Now, the easiest answer would be to combine this type of data with other markers from the dog. For example, if you think about that genetics play a very important role in how metabolism occurs in a healthy organism or a non-healthy organism, then you would maybe be interested in looking at gene copy numbers and seeing just how many genes are in one set of dogs compared to the other. Another would be looking at the microbiome, because it seems that a lot of metabolic processes actually begin with the gut microbiome breaking down food and converting it, and then before it actually enters the blood system. So combining it with other biomarkers would be is very interesting. Another avenue of or approach would be to see how it correlates with other phenotypic markers. For example, we were looking at a chronic disease state, and we were looking at both healthy individuals and diseased individuals. And we were looking at, does the diet actually modulate this disease state in any way? And so there are official criteria for diagnosing how severe a chronic disease can be. And there are basically, you can take these type of phenotypic criteria and see, are there any correlations with the changes that you can see in metabolite profiles. So, for example, we're specifically looking at a disease called canine atopic dermatitis, which is a common chronic skin disease. And it has a set of validated criteria given in points for how severe the disease is. So over time, you can, over the diet intervention period, you can say, okay, this dog was eating this diet and had a score for this disease of this number, and then it changed to this number over the time that it was eating this diet. And so you can also look at it from that point and get a lot of information. It's quite useful. The third would be just to see, well, what are the metabolite concentrations and what is considered healthy and unhealthy. You can just check it with reference values as well. So ideally, combining all three would be what you could do, but already just combining the latter two of seeing how it correlates with disease severity, and also how it correlates with reference values that have been established already beforehand are the best avenues for interpreting it.
Okay, so do you have some published results, or do you only have, at this point, the kinds of results that you can't really talk about too much?
So I've presented at some conferences, I've presented posters of my work, but the article is currently being finished up and is being submitted for publication soon. We have a couple of articles that are dealing with metabolomics. One that I'm finishing up now is interested in only looking at what information we can gain from looking at metabolic profiles. The other one is much harder to accomplish, but I think much more interesting, where we're going to take information we know about metabolic profiles and then tie that into other biomarkers. So we can look at gene expression analysis, and we can look at blood hematology values, classic clinical markers, as well as microbiota and lifestyle factors. So what we can say now is what I've noticed, and that's what I've noticed from just looking at metabolic profiling without looking at other markers. So if we look at the metabolism of a dog from the points of metabolic profiling of the metabolites that we've been looking at, there's a couple of things that we can see. First of all, the energy metabolism of these dogs change quite radically.
Okay, what does that mean?
Okay, so classically, a mammal creates energy from a molecule called glucose. It's a sugar molecule. And it's broken down by a process called glycolysis to then feed mitochondria with substrate for energy. And what the mitochondria can do is that it can actually get energy preferentially from glucose, but if glucose is not around, it can actually switch over and get energy from other sources. Now, glucose is most easily bioavailable to the body by breaking down starch and breaking down carbohydrates. And so one of our diets has a lot of carbohydrates in it, and then the other one doesn't. The other one actually has 0 carbohydrates in it, which means that glucose is being produced, but it's being produced endogenously. The body is actually converting the nutrients and the nutrition in the food into glucose without there being actually any glucose.
Like maybe protein?
Yeah, so fat actually.
Okay.
Fat would be preferential. So, but protein, it is possible to convert protein into glucose as well. Yeah. But preferentially, gluconeogenesis is the process of converting a fat molecule or a fatty acid into glucose.
Okay, I thought that was protein.
Oh, well, it's actually both. I mean, both can be converted into glucose, and it depends on the amino acid. Some are more readily converted into glucose than others. But it's interesting that you can say, okay, well, the energy metabolism basically in a dog relies around having a very tightly regulated amount of glucose available, but then in case there isn't enough glucose, having enough of other substrates available. And the most easily broken down substrate for a dog besides glucose is fatty acids. And it seems that a dog is very readily able to break down fats into energy. And there seems to be evidence that dogs have been doing this for a very long time. So energy metabolism is interesting. And what I think is interesting is that if you look at, actually, we looked at the glucose levels, blood glucose in both dogs, and they're remarkably similar. So we can know that even if one diet has way more carbohydrate than the other, then the glucose metabolism stays quite similar. There isn't any significant difference between the two. Okay. Another thing that you can look at is, okay, so we were looking at energy metabolism. Another thing to point out is that you can actually, we were able to see that the dogs that were switched to the non-processed diet that didn't have any carbohydrate actually fundamentally switched their metabolism to prefer something called the ribose phosphate pathway, or the pentose phosphate pathway, which is a clear indicator that they are actively breaking down fats for energy to create glucose and other energy-rich molecules for mitochondria. So we were able to see that, which is, well, obvious at the end of the day. And then the other thing that we can see is, in our metabolomic profile, we had a lot of amino acids and nucleotides. So we can sort of posit what the differences in concentrations of these amino acids are, because single amino acids that are traveling around the body are traditionally signaling molecules. You don't really find them just flowing around waiting to be built into protein, right? So you can, or it's been shown that many of these free amino acids going around blood serum have signaling purposes, and these signaling purposes mainly center around dealing with stress and dealing with inflammation. So those types of things are quite important. Amino acids also, of course, have a central role in energy metabolism.
Okay. What about the health or disease markers of the dogs, or is it too early to talk about that?
Right. So if you want to look at disease markers from a purely metabolite point of view, so you're looking at only the metabolites that we've included in our targeted metabolic profile, then there hasn't been enough data published previously about what the reference values should be for these in a dog for us to be able to conclude that this is for sure a biomarker of disease, or this is for sure a biomarker of a disease progressing and becoming worse. So there isn't enough evidence for us to directly say that, oh, okay, well, there's clearly like way too much of this biomarker, so that's why this dog is going to get sicker and sicker. So we can't say that because there simply hasn't been a study that has been able to give us clear answers regarding that. But we can indirectly look at how biomarkers and elevated concentrations of certain metabolites have been linked to chronic disease states. What's interesting is that although very little has been published regarding dogs, dogs and humans actually share many metabolic pathways. And so there's plenty that we can learn by studying the same disease that affects both dogs and humans by looking at human data and it educating us and giving us a better idea of what we should be looking for next, or where future exploration should go.
Okay. Can you directly see that if you feed this kind of diet to this kind of group of dogs, that it affects their health in a certain way?
Right. So, like I said, we cannot do it directly, but we can get indicators that are indirect. For example, there's a few classic markers for dealing with chronic stress. And we know there are some mechanisms for dealing with chronic stress that involve, for example, epigenetic programming. If, like we were talking about at the beginning of our conversation, you have an animal that's environment has been changed radically, then you will see that epigenetic programming will be kicked up in gear. You will see that the body is starting to have to express different genes to adapt to a new environment. What's interesting is that you can actually look at the amino acids that have central roles in regulating this process and get some kind of idea that, okay, well, we can't say that this diet is causing this disease to worsen, but we can say that there seems to be indications that this diet is causing a much higher concentration of these certain markers that are classic for increased epigenetic programming. And so that doesn't give us an exact answer, but it does raise the question that why does that happen? And it also does give us the motivation to study it further. So, for example, there is you can think of it as, well, especially in the state of a chronic disease, that you can approach a lot of chronic disease states that are not purely genetic in nature as being sort of a constant stressor on the body. It's just sort of giving the body seems to be stressed. And this is a state that can induce epigenetic programming, because if it's stressed, there's something that is not going as efficiently as possible, so the body tries to correct for it. Now, if you see that there are the metabolites required to allow for this type of reprogramming to occur being in much higher concentrations in the ultra-processed diet, then you can indirectly say, well, this seems to be an indication that there's some type of a stressor happening in this diet. And we did that by looking at both healthy individuals and how they responded to diet, and by looking at chronically diseased dogs and how they respond to diet. And what's really interesting is if you try to look at the entire metabolic profile and you try to classify them, these different dog groups, you have 4 groups. You have the non-processed diet groups that are healthy and diseased, and then you have the dogs that are eating the ultra-processed diet that are diseased and healthy. So you have 4 groups, right? And what we found was that the healthy individuals are very difficult to discern against. It's very difficult to look at, to say, well, this dog was healthy and this dog, it's very difficult to say that this dog was eating the ultra-processed diet and this dog was eating the raw food diet. But then when we look at the diseased individuals, it's very obvious this dog was eating the raw food diet and this dog was eating the unprocessed diet. So it seems that the underlying disease state modulates the outcome of how a dog responds to food. And that's, I think, very interesting. Like,